Good morning. Let's get started here today. My name is Chris Shibutani. I'm a member of the biotech research team here at Cowen. Welcome to day 3 of our conference. On Monday morning, we had a panel of experts of the immuno-oncology field looking at novel targets, and there continues to be clear evidence from our survey of experts in the field of tremendous interest in activators of the innate immune system, of the abscopal effects that we can see. And as the science has evolved, the company has continued to sharpen their focus. Investors, I think, have a unique opportunity at this point in the year as we'll actually get to see some data readouts on both of the focus programs, in particular for STING, but also for additional programs that the company is continuing to nurture. Here to give us an update, Steve from Aduro.
Thank you.
Thank you.
Okay, thanks, Chris. I am Stephen Isaacs. I'm the chairman and CEO of Aduro, and pleased to be here at the 39th annual healthcare conference of Cowen and Company to tell you all about what Aduro is doing. So, as you would expect, I'm going to be making some forward-looking statements during the course of the presentation, and if you have any questions about any of those statements, I would refer you to our filed documentation with the U.S. Securities and Exchange Commission. So now, getting on with what's really important, I want to start with our Aduro corporate highlights. Aduro works primarily in 2 different areas. One is in STING biology. STING is an acronym that stands for Stimulator of Interferon Genes. And the second area we spend our time in is in APRIL biology. So APRIL stands for a proliferation reducing ligand. So in STING, we have a very important collaboration with Novartis that we've had for the past 3 years on STING agonists. So agonists stimulate STING in a way that generates innate and adaptive immunity to fight different types of cancer. The particular compound we're developing at the present time is called ADU-S100. It's sort of a custom cyclic dinucleotide that Aduro made several years ago, and we're using this in different types of tumors that we do a direct inject approach of. We have some very encouraging preliminary signs of clinical activity. We reported the status of the program and the monotherapy trial and a little bit about the combination trial at SITC in November. And we'll be updating these results, as Chris said in his introductory remarks, either at ASCO or ESMO this year. On the other side of the STING coin, you want to stimulate STING for oncology, but if STING is overstimulated, it results in autoimmune and inflammatory disease. And so we have another program to shut STING down by generating small molecules along the cGAS-STING pathway as inhibitors. And that's the deal that we did with Lilly toward the end of last year. So we do view ourselves to be very much experts in the STING pathway. We've been involved at the basic research level and developmentally for several years. We've published extensively and feel that we have a lead. And our most recent report came out in Cell Reports just a couple of months ago, showing that low-dose STING agonism results in robust innate and T-cell immunity, adaptive T-cell immunity from the injected lesions, and this is very important in simulating both adaptive and innate immunity. So APRIL, again, APRIL stands for a proliferation reducing ligand. This is a wholly owned asset that is unpartnered at the present time. We have an anti-APRIL antibody we call BION-1301. This antibody is in clinical development for multiple myeloma, and it's also being pursued in a new indication, a non-oncology indication called IgA nephropathy that we're very excited about. We also have a leadership position in the APRIL pathway, work very closely here at the Dana-Farber with Ken Anderson, and had a recent publication in Leukemia that highlights some of the aspects of the research and development activity that we've pursued. Strong financially, $278 million at the end of Q4 of last year, and that provides operating capital well into 2022 and does not include any of the significant upcoming milestones we have during that period of time from our 3 corporate partners. So this is a little closer look at our pipeline. So in STING, again, stimulator of interferon genes, we have 4 clinical trials, 3 of which are active. The most advanced is the monotherapy dose escalation with ADU-S100. This is in multiple tumors. We reported out the first 7 dose groups. We'll be completing that trial soon, and we'll have a full report either at ASCO or ESMO this year. The next step was to combine ADU-S100 with an anti-PD-1. And in the first trial, we're using spartalizumab. That is Novartis's version of anti-PD-1. That, again, is in the same group of multiple tumors. It's progressing very nicely. We gave some indication of the initial results, again, at SITC in November. And then more recently, we went into a more defined population, relapsed refractory melanoma, with ADUS-100 in combination with ipilimumab or anti-CTLA-4. And we announced the first patient treated just a couple of months ago, and that trial is proceeding nicely. And then we're going to be starting a trial in a head and neck setting, earlier head and neck, and that'll be ADUS-100 in combination with an approved anti-PD-1. And then again, on the inhibitory side, the other side of STING antagonism along the cGAS-STING pathway, that remains in discovery. We just started that with Lilly and hope to be moving that into preclinical in the near future. So then for April, we have BION-1301. This is an anti-APRIL antibody useful in multiple myeloma. That's in a phase 1/2 study. And then we'll be going into the IgA nephropathy setting. That's an autoimmune disease in the first half of this year. So that's kind of the big picture of what we're doing. Now I'm going to get a little bit more specific in the two focus areas of STING and APRIL. So starting with ADUS-100, which is a STING agonist. The STING receptor, the STING pathway, really plays a very critical role in activation of tumor immunity. And so basically when a naturally occurring cyclic dinucleotide called cGAMP binds to STING, it sets up a cascade of events that generates type 1 interferons, of which interferon beta is the main player. And this stimulates interferon genes, and it's a very critical component of both innate immunity, and it leads to a very broad adaptive immune response that can affect tumors in an abscopal effect manner. STING activation is required for the rejection of cancer in various mouse models. We showed that very clearly. If you use STING knockout mice, this pathway eliminates the ability to generate an immune response, and this differentiates us from the various approaches with the TLRs. So we made a molecule called ADUS-100. Novartis calls it MIW-815. It's a first-in-class STING agonist, very, very potent. We demonstrated very strong preclinical anti-tumor activity in a mouse model, particularly lethal B16 melanoma mouse model. The results were generating innate immunity, leading to an adaptive immune response with a very strong abscopal effect. And this led to sort of a groundbreaking deal with Novartis, where they paid $250 million up front based on preclinical data, and led to a co-development and co-commercialization collaboration. So ADUS-100 has been in the clinic now for a while in a monotherapy dose escalation and now in a combination trial with spartalizumab. And we have some very encouraging preliminary clinical signals in these ongoing early phase trials. And again, we'll be reporting more about that in the course of 2019. So we're advancing ADUS-100 in clinical development in sort of two different ways. You can think of it as two different patient populations, both through intratumoral injection. On the left, those two boxes are intratumoral injection in a very heterogeneous, heavily pretreated set of patients. And then on the right, where we're going now, is based on what we learn in these heterogeneous populations, is to apply that knowledge in more homogeneous patient populations, such as relapsed refractory melanoma and earlier line head and neck. So the monotherapy trial with ADUS-100 was a first-in-human proof of mechanism study, obviously looking at safety, but for hints of clinical activity and a full biomarker analysis. We're going into metastatic solid tumors and lymphomas, and then we're using that same population to do a proof of mechanism or proof of concept trial, I should say, in combination with spartalizumab, again in these heterogeneous patients. And that's being followed by the ipilimumab trial, the combination of ADUS-100 with anti-CTLA-4. And this is to demonstrate combination activity in the more defined population of relapsed refractory melanoma. You know, that's really where all the blue sky is where checkpoints either stop working or don't work at all. And then we want to extend that concept into an earlier line head and neck population. So this is the monotherapy study. We reported this data at SITC back in November. So this is a dose escalation trial. We started very low at 50 micrograms, and then we doubled up through 3,200 micrograms, so that's 7 dose groups, and we talked about these results in 41 patients at SITC. We went into cutaneously accessible treatment refractory advanced or metastatic solid tumors or lymphomas. Again, that was a patient population. And the primary objective was safety and tolerability, but of course you want to take a look at what efficacy might look like and what the early hints might be. In terms of the patient population, nothing remarkable. We'll point out that about half the patients in this study, half the 41 patients through the 7 dose groups, had prior checkpoint inhibitor therapy, which is very important, and about 80% or so had at least 2 prior regimens, and some of them had 9, 10, 11 prior regimens. And importantly, the number of tumor types we looked at in this group was 22. No significant DLTs were observed in the study. No patients were discontinued due to an AE. The FDA was quite worried because of the potency of the agonist that cytokine storm might be a problem, but that turned out not to be the case at all. And so it took quite a while to escalate through all these dose groups. These are the results, a little hard to see. Basically, out of 41 patients, we saw 2 partial responses, saw one in a Merkel cell carcinoma and another in a parotid gland head and neck cancer. And as of the time we reported this data at SITC, there were 4 patients with ongoing responses. So we had 2 PRs and 11 patients who stabilized and had stable disease. We considered this a good result in this particular advanced heterogeneous patient population. The safety profile was very good. Basically, we didn't see any DLTs that caused patients to come off study. It's really quite an innocuous therapy. There were preliminary signs of clinical and biomarker activity. Again, 2 of the 41 patients had partial responses, and one of those patients had a prior anti-PD-1 therapy, and 11 patients achieved the status of stable disease. In terms of target engagement, we did observe the generation of some of the key systemic cytokines that you would expect to result from stimulation of STING, including IL-6, MCP-1, and importantly, interferon beta, and this did indicate engagement of the target and activation of the STING pathway. So in the phase 1b, this is a combination of ADU-S100 and spartalizumab. Basically, it's in the same patient population and the same approach to cutaneously accessible treatment refractory advanced or metastatic solid tumors or lymphomas, so the same as we did in the monotherapy dose escalation. Again, the primary objective was safety and tolerability. This time we took a look at 2 dose groups. The first was similar to what we did in the monotherapy, so the patients received ADU-S100 on days 1, 8, and 15 of a 28-day cycle, and that was repeated until progression, and they got the PDR-001, the anti-PD-1, on day 1. In the second dose group, there was a single dose of ADU-S100 on day 1. So this is for purposes of dose selection and going into dose expansion studies in these relapsed refractory populations in melanoma and earlier line head and neck. And we are doing some backfilling now of certain patient groups where the responses are more predominant. So it's really sort of working our way through this to see which indications look good for advancing into expansion. So this is what we said at SITC, and again, we'll be updating this at either ASCO or ESMO. The dose escalation was in 50 patients at the time. This was back in November, and this was with full dose anti-PD-1 or PDR-001 and increasing dosing of intratumoral ADU-S100. No DLTs were reported. The safety looked very good, consistent with the monotherapy study. In terms of responses, 2 patients had previously demonstrated responses to checkpoint inhibitor therapy alone. And again, this is very important. The population of relapsed refractory patients who do respond to the therapy post ADU-S100. We did indeed see reduced tumor volume in both the injected and non-injected lesions in some patients. And of course, this isn't about the non-injected lesions. This is about micrometastases. So that's exactly what we wanted to see. And several patients remained on study for greater than 6 months. And again, we'll be expanding upon these results, the biomarker results, the response results, et cetera, later in the year. So in terms of APRIL, BION-1301, this is an anti-APRIL antibody. It's fully blocking, fully novel, first-in-class antibody that we made. So APRIL is made in the bone marrow. It's a soluble factor, and it binds to BCMA and TOSI, for instance, in multiple myeloma cells, and that induces signaling that results in negative outcomes. So if APRIL can be blocked, this is a distinct approach to inhibit both BCMA and TOSI effects, so it creates this potential immunomodulatory opportunity. So BION-1301, again, a first-in-class monoclonal antibody, which blocks APRIL by binding to both BCMA and TOSI receptors and preventing that from occurring. And it's very well tolerated in preclinical studies and now in our ongoing clinical studies in multiple myeloma where it's implicated. And then also we see it being used in IgA nephropathy and in other indications. So in multiple myeloma, the problem is when APRIL binds to BCMA, it induces proliferation and drug resistance, and when it binds to TOSI, it sets up this immunosuppressive phenotype. So again, if you intercept the APRIL before that happens, we think we can reverse those effects. We've demonstrated this as a single-agent activity, inhibiting myeloma cells, and work with Ken Anderson at the Farber. We published those results, and so now we're looking at this in the clinical setting. So this is our phase 1/2 BION-1301 study in multiple myeloma. This trial is ongoing. So the patients are relapsed refractory in multiple myeloma, and their disease has progressed after at least 3 prior systemic therapies. The objective is safety, of course, and to establish a recommended dose and schedule for expansion. So the patients receive BION-1301 on days 1 and 15 of a 28-day cycle, and that's repeated. We're also looking at some additional dosing schedules now and trying to determine the PK and PD to go on and set the dose and the schedule for the phase 2. So the next indication we're very interested in is IgA nephropathy. So this is not oncology. This is an autoimmune disease, very interesting disease and somewhat of a neglected disease with a fairly significant prevalence. So IgA nephropathy results from a sort of a mutant IgA. So when galactose-deficient IgA occurs, and this is a function of high APRIL concentration, basically the body responds in an autoimmune fashion, and it creates an anti-galactose-deficient IgA antibody. And so, of course, the antibody combines with the antigen. It forms these complexes. The complexes circulate, end up in the kidney, and basically leads to inflammation and ultimately renal damage and end-stage renal disease. So the disease overview is that the patients produce galactose-deficient IgA, then they get an antibody to that galactose-deficient IgA. That results in immune complexes causing inflammation. The symptomatology is proteinuria and microscopic hematuria. And the FDA, incidentally, has gone away from end-stage renal disease as the endpoint and will now allow proteinuria, which makes this a feasible study to do in a reasonable amount of time. The next stage is GFR, so the filtration rate breaks down. This leads to the need for transplant, which doesn't really solve the problem because it just gives a new kidney for the same deposits to form in, so it delays the disease, but it doesn't stop it. And then, of course, dialysis, and this can be a fatal syndrome. IgA nephropathy has no current cure. There doesn't seem to be a lot of attention being paid to it. It's a significant disease, especially in East Asia. The true global prevalence isn't known, but it is known that it's very significant. It requires a kidney biopsy to confirm diagnostically. And it is known that something like 15 to 25% of the patients progress to end-stage renal disease within about 10 years. And the slide says 20 to 30% after 20, but more recent publications suggest this number could be as high as 50%. So we do see this as a real opportunity, and we're getting progressively excited about it. I was just in Japan last week talking to several potential corporate partners. Not that we would partner at this point, but there is an awful lot of interest in it. So what we want to do is use anti-APRIL to knock down the mutant IgA. The upper left-hand panel is a transgenic mouse with high levels of APRIL showing high levels correspondingly of IgA that's produced. This is a well-known phenomena in people with the syndrome as well. The bottom panel shows that if you use the mouse version of BION 1301, you can knock these IgA levels down very significantly. And then the right-hand panel shows the same result in primates. So you can take primates and you can see the reduction in IgA in a dose-dependent manner, which is exactly what we want to see. And now we're going into the human population to see, hopefully, exactly these same results. So the first-in-human study of BION 1301 in IgA nephropathy is planned. Although we do have a very good safety profile in multiple myeloma patients, we want to get kind of the baseline in normal human volunteers for safety. So we're doing a double-blind, placebo-controlled single ascending dose with up to 5 doses to set the safety parameters. The trial really is to assess safety both in healthy volunteers and in IgAN patients, and then use PK/PD, those intermingled relationships in these patients to set the dose and then to establish proof of mechanism. So the second part of the study is multiple ascending doses, and we can overlap that with the first, kind of building up in the safety that we observe in the single ascending dose, and then integrate in the actual IgAN patients about halfway through the multiple ascending dose study. So that's what we're doing in STING in April at a high level. We can answer, whatever questions you may have in the breakout session. In terms of the business overview, we have partnered with Novartis, with Lilly, and with Merck. These are very significant relationships. The Novartis relationship I mentioned early on in my remarks. This is around ADU S100 agonism of STING. Actually, any STING agonist is under the agreement. It's a $750 million deal, $250 million up front, another $500 million in development milestones, not the typical commercial milestones. So this sort of pays our way in paying for half the R&D. Co-development and co-commercialization is the basis for the agreement. The way it works out is that we pay 38 cents of every R&D dollar, very good deal for us. We have control in the U.S., we'll lead commercialization, pricing, etc., and we'll split profits 50-50. And in the European and Japanese markets, we'll split profits 45-55, with Aduro getting the smaller share, and then a high teens royalty rest of world. So we're very, very happy with that. And we're very happy with Novartis. We have a major joint steering committee meeting coming up on Thursday and Friday, and the relationship is strong. I spend quite a bit of time with Jay Bradner here in Cambridge, talking about his enthusiasm for the program. We inked the deal with Lilly back in December on the other side of the STING coin for STING antagonists. This is for the CGAS-STING pathway inhibitor program to address autoimmune and inflammatory disease, working with A.J. Norella, who was brought from Amgen to Lilly to build this franchise that will be a very significant portion of their revenues in years to come. It was a much smaller deal up front, but it is $620 million per product potential in terms of development and commercial milestones. Lilly will have full responsibility for global commercialization. We will get a single to low double-digit royalty and, of course, milestones. And we do have the option to co-fund clinical development further out in exchange for significantly increased royalties, which we look forward to potentially taking advantage of. And then finally with Merck, we have an anti-CD27 agonist that we licensed to them for oncology. It was a $447 million deal. Collected a significant portion of that in terms of milestones. It's a global license, and there are multiple single-digit to low teens royalties. And Merck just issued their annual report, and they're quite excited about this program. So we're fortunately in a very strong financial position. It's where we want to be. We always like to keep the balance sheet healthy. We had cash and cash equivalents at the end of 2018 of about $278 million. R&D last year was about $76 million, and G&A about $36 million. Those numbers will drop. We've slimmed the company a bit as we focus specifically on STING and on APRIL, and we're looking to out-license some of the other programs that we've developed, and about 80 million shares outstanding. In terms of patents, we have global rights, including in-license patents, to about 150 composition and matter and method patents, and another 300 pending applications. And these expire in the mid-20s to the late 30s, so we have a very good IP picket fence around all that we do. And finally, this current cash equivalents, and marketable securities do provide operating capital well into 2022, and that is without the very significant milestones that we do anticipate receiving from Novartis, from Lilly, and from Merck. So finally, this is our upcoming anticipated milestone slide. So again, focusing on STING in April. So for STING, the first thing we wanted to do this year is start the ipilimumab ADU-S100 combination. That was accomplished, and that trial is underway. This is phase 1 dose escalation, looking at the more defined relapsed refractory melanoma population. And then with spartalizumab ADU-S100, that's our combination trial that I've commented on, and so we do anticipate sort of full disclosure on the biomarker and clinical response data, either at ASCO or ESMO. We'll see, the response to the abstracts. Then we want to go into the the phase 1 dose expansion study in the spartalizumab ADU-S100 combination in several different indications. Those will be starting in the second half. And then initiating our earlier line head and neck cancer study with ADU-S100 and an approved anti-PD-1, which we feel is very important from a regulatory point of view. And then in the April program, initiating our IgAN study that I just described will occur in the first half. And then we'll be reporting out some of the phase 1 multiple myeloma dose escalation results and exploring the initiation of phase 2 in the second half. So that's the story, and I think we have a breakout session, but I'm not sure where. Yep, sure. And introduce Andrea Van Elseth. He's our Chief Scientific Officer. They can take some questions. Yes, Chris.
Why is the IgA raised in IgA nephropathy?
So it appears to be stimulated by APRIL. APRIL stimulates the plasma cells that produce the IgA in the first place, and so there's a correlation between high levels of IgA and high levels of APRIL and high levels of IgA, and we've seen this in mice, and we've also it's reported in humans. We haven't seen it directly. So the idea is that if you can inhibit APRIL, you can knock the IgA down. We've seen this in mice. We've seen this in primates, and hopefully we'll see this in humans. And
is it a single clone of IgA?
So I'll let Andrea address that.
The disease is caused by the autoantibody reaction improperly glycosylated or glycosylated. So the glycosylation antibodies are seen as an autoantigen. In some patients or some healthy individuals, it leads to the autoantigen response, and then you get this downstream deposition of immune complexes, and that actually causes the disease. So it's not so much that there is a lot of IgA being produced, but it's the immune response to the galactose-deficient species that actually causes disease. And in fact, you and I also probably have a fair amount of production of galactose-deficient IgA, but apparently we don't mount the immune response to that, so nobody really knows yet what makes the difference between patients and healthy individuals. But what's important is that the APRIL affects two aspects of the disease. One is the production of the galactose-deficient IgA, but it also inhibits the autoantibody response. So we think there are two key elements that we think are associated with the disease response.
Chris?
Great. Actually, that was all.
Okay. Sure. Thank you.